This PowerPoint covers the function of the pancreatic hormones such as insulin, glucagon, and somatostatin, and the parathyroid hormone, and calcitonin, and vitamin D. The pancreas is located deep to the stomach and inferior to the liver. Its head rests snugly in the concave curve of the duodenum, and its tail tickles the spleen. The pancreas functions as both an exocrine gland and an endocrine gland. The endocrine glands are located at the tail of the pancreas. The pancreas produces three hormones: insulin from beta cells, glucagon from alpha cells, and somatostatin from delta cells. All three of these hormones are protein hormones, which is the largest classification of hormones in the body. They are water soluble and are able to travel freely throughout the circulatory system. The receptors are located on the cell membranes of target organs or target tissues, and they can be stored in vesicles within the endocrine gland that produces them. The secretion of insulin and glucagon is regulated by negative feedback from blood levels of glucose. High levels of glucose stimulate insulin secretion, while low levels of glucose stimulate glucagon secretion. Insulin is synthesized in the islets of Langerhans beta cells. There are several steps to this process, but we're going to just cover the three main ones. Initially, amino acids are linked together in the ribosomes that are on the endoplasmic reticulum, known as rough ER. They will link together a pre-proinsulin polypeptide. This pre-proinsulin will then go into the cisternae clefts of the endoplasmic reticulum, where it is cleaved and becomes proinsulin. The proinsulin is then taken up by the Golgi apparatus, where it is cleaved again and repackaged into insulin and a C-peptide. Mechanism of insulin secretion. GLUT2 transporters facilitate glucose uptake into beta cells. Once in the cell, mitochondria will break down the glucose to make ATP. Increased intracellular ATP closes ATP-dependent potassium channels, which leads to the depolarization of the beta cell membrane. This depolarization will then open voltage-gated calcium channels. This influx of calcium will cause the exocytosis of insulin-containing vesicles. Beta cells have gap junctions linking each beta cell to the other. This allows synchronization of depolarization and allowing the beta cells to release their insulin contents all at the same time. Insulin is released on a pulsating rhythm about every 5 minutes. I believe I've mentioned before that all hormones are released on a pulsating rhythm. Because insulin is typically released more than once within a 24-hour period, it is considered part of the ultradian rhythm. There are two stages of insulin secretion: acute and delayed. The acute or immediate stage is stimulated by acute elevation of blood glucose, say, for example, after a carbohydrate-high meal. Beta cells will then dump the pre-made stored insulin, because if you remember, protein hormones can be stored in vesicles within the cell that produces them. This will increase plasma concentration of insulin tenfold within 3 to 5 minutes. Then in 5 minutes, the insulin will drop to half its normal levels because it's quickly used up. Then in 10 minutes or 10 minutes later, the insulin levels return back to normal. And that's because it takes that long for the beta cells to produce more insulin. This acute stage, is actually absent with type 2 diabetics. The delayed or sustained stage of insulin secretion occurs about 15 minutes after the acute stage. Insulin secretion rises a second time, usually at a higher rate than the initial secretion. This can last 2 to 3 hours depending on how many carbohydrates was consumed and how much is circulating in the bloodstream. Functions of insulin. The most notable function of insulin is to promote glucose uptake, and it also promotes intracellular storage of glucose. It affects almost all body tissues, but especially muscle and adipose tissue. It will stimulate GLUT4 receptors or transporters within the cells that allows the cells to uptake a large amount of the glucose at one time. Without insulin, glucose transporters will only work at about 25% of their capacity. Another function of insulin is to promote amino acid uptake by cells, which will stimulate protein synthesis. Increased intracellular amino acids will lead to increased activity and number of ribosomes. Insulin is said to turn on ribosomes. This increase of intracellular amino acids will also lead to the increased storage of amino acid or storage of proteins. Another function of insulin is that it reduces lysosome normal digestion of proteins, which will inhibit protein catabolism. So insulin also functions as a protein sparer. Another function of insulin is that it facilitates growth hormone effects on skeletal muscles, such as it promotes amino acid uptake just like growth hormone does. In fact, insulin's effect on amino acids is almost identical to that of growth hormone. Another function of insulin is that it promotes fat synthesis and storage. It does this by inhibiting lipase, fat breakdown, which also leads to inhibiting lipid catabolism. Another function of insulin is that it promotes liver glycogenesis, the production and storage of glycogen. It does this by increasing the activity of enzymes that promote glycogen synthesis. Specifically, it promotes the activity of glycogen synthase. Insulin affects the liver in several ways. One of the ways is that it promotes conversion of excess glucose into fat. The liver stops producing glycogen after 5 to 6% concentration is stored because the liver has a maximum saturation point. The remaining or excess glucose is then converted into low-density lipoproteins. These low-density lipoproteins are transported to adipose tissue for storage. Another effect insulin has on the liver is that it decreases the rate of liver gluconeogenesis. It does this by inhibiting the activity of enzymes necessary for gluconeogenesis. This is one of the primary causes of high glucose levels in type 2 diabetics. Hepatic cells lose sensitivity to insulin, and then it's no longer able to inhibit the enzymes that the liver uses for gluconeogenesis, and the liver just continues to pump out glucose into the system. This is actually the process that the medication metformin works on for type 2 diabetics. It helps to activate the enzymes necessary to inhibit gluconeogenesis. The insulin will use the liver as a major glucose buffer in the body by promoting it to store glucose as needed and then stimulating it to release glucose or break down glycogen as needed to maintain blood glucose levels. Lack of insulin or insensitivity to insulin will stop synthesis and storage of proteins. You'll have a decrease of intracellular amino acids and therefore decrease in number and activity of ribosomes. This will stimulate protein catabolism, causing cells to dump amino acids into circulation. This increase in serum amino acids will also promote gluconeogenesis, and the enzymes required to promote or facilitate gluconeogenesis are no longer inhibited by the insulin. Again, a problem with type 2 diabetics. The lack of insulin or insensitivity to insulin will no longer inhibit lipase, and it will actually strongly stimulate the activity of lipase, which will liberate stored triglycerides, releasing large quantities of fatty acids into circulation. This can lead to atherosclerosis in people with serious type 1 or type 2 diabetes mellitus. The lack of insulin or insensitivity to insulin causes ketogenic effects. Long-term absence of insulin, as in the case of type 1 diabetes and sometimes in type 2, leads to excess usage of fats for energy production. This causes excessive amounts of acetoacetic acid produced by the liver. Acetoacetic acid is produced at a faster rate than it can be metabolized, causing ketosis and leading to a diabetic coma. Diabetes mellitus. You have had plenty of information on this condition before you've reached this class, so I'm going to be very brief. Type 1, also known as insulin-dependent or juvenile diabetes, is a result of diminished or no insulin production secretion at all. This can be due to damage of beta cells. Beta cells can be damaged by a viral infection or an autoimmune disorder, and genetics can play a role into making the beta cells more susceptible to this type of destruction. The other type is type 2, also known as non-insulin dependent or adult onset diabetes. This is due to a decreased sensitivity of target cells to insulin. They become insulin resistant. We did speak about specifically how the liver hepatic cells become resistant, and it cannot prevent the liver from producing glucose in gluconeogenesis. Other factors that can cause insulin resistance is polycystic ovarian syndrome, hypersecretion of cortisol, which we'll cover later on, and hypersecretion of growth hormone, which we have already covered. We do have tissues and organs in our bodies that don't need insulin. They do not require glucose transporters to uptake the glucose. And these are the brain, liver, kidneys, gonads, epithelial tissues, and red blood cells. Somatostatin is produced by islets of Langerhans delta cells. Secretion is stimulated by ingesting food, and its principal role is to extend digestion time to allow maximum absorption of nutrients. Somatostatin acts locally within the islet cells as a paracrine. It inhibits secretion of both insulin and glucagon. It decreases the motility of the stomach, duodenum, and gallbladder. It decreases secretion and absorption of nutrients in the gastrointestinal tract to extend the period over time which these foods and nutrients are assimilated into the blood. Somatostatin has the same chemical makeup as growth hormone inhibitory hormone, so it will also inhibit growth hormone while it is active. The primary function of glucagon is to increase blood glucose levels. It does this by causing the liver to break down glycogen and to produce glucose through gluconeogenesis. One of the ways it promotes that is by increasing the rate of amino acid uptake by hematocytes, which is one of the substrates necessary for gluconeogenesis. Glucagon also activates lipase, which will mobilize fatty acids, making them available for tissues to use for ATP production while glucose levels are low. It'll also inhibit liver's production of triglycerides, so it'll prevent also fat storage while it's active. Norepinephrine can act like glucagon. By that, it stimulates the breakdown of glycogen in skeletal muscle. Humoral regulation of serum calcium. The body regulates blood levels of calcium by secreting the appropriate amount of parathyroid hormone, vitamin D, and calcitonin when necessary. Calcitonin is produced by the thyroid gland. The thyroid gland is located in the area of the throat, just below the larynx and anterior to the trachea. It is made up of follicles and interstitial space or tissue within the follicles. Calcitonin is produced by parafollicular cells or C cells or clear cells within that interstitial space between the thyroid follicles. Calcitonin is released by the thyroid gland when serum calcium levels are high. It decreases the activity of osteoclast cells, which reabsorb bone, and increases the activity of osteoblast cells, which increase bone matrix. Calcitonin primarily promotes building or strengthening of bone. It has a very weak effect in the adult because of the slow rate of bone remodeling, but a very strong effect in children who have a rapid rate of bone remodeling. The parathyroid hormone is produced by the parathyroid gland, which are four little oval-shaped structures located on the posterior aspect of the thyroid gland. It has an opposite effect of calcitonin in that it promotes destruction or reabsorption of bone. Parathyroid hormone is released by the parathyroid gland when serum calcium levels are low. It increases the activity of osteoclasts and decreases the activity of osteoblasts. It also stimulates the kidneys to reabsorb calcium out of the filtrate, and it stimulates the kidneys to activate vitamin D, which is necessary to increase absorption of calcium from the intestines. Vitamin D is a steroid hormone synthesized from cholesterol, initially in the skin, and then it goes through several step processes that you learn from Dr. Zoe in biochemistry. All we're going to focus on here is that the fact that it is a steroid hormone and that it is required for the absorption of calcium from the intestines.